Good day, everybody. It is Faye from the 𝐂𝐞𝐿 Review Center. And today we are gonna talk about chromatin packaging and DNA supercoiling. So how long do you think DNA in a single human cell is? Well, the answer is almost 2 meters long, and yet it fits inside a nucleus only 10 micrometers wide. How? Well, the answer lies in supercoiling and packaging. Good. So what is DNA supercoiling? Well, DNA supercoiling is the twisting of DNA to make it more compact, like twisting a rubber band or a telephone cord. This coiling the DNA fit in small spaces while staying functional. The quick one. Have you ever twisted a rope so much that it coils back on its own? That is the same idea. DNA does this naturally to reduce its overall length. There are two types of DNA supercoiling, the positive and negative supercoiling. Positive supercoiling, DNA is overwound, making it tighter and harder to separate. Negative supercoiling, DNA is underwound, making it looser and easier to access. Cells usually maintain negative supercoiling, especially during replication and transcription. It makes unzipping the double helix much easier for enzymes. Here's a quick one. Which do you think helps with DNA reading? Is it positive supercoiling or negative supercoiling? Well, the answer is negative supercoiling. Supercoiling is controlled by special enzymes called topoisomerases. Topoisomerase 1 and topoisomerase 2. Topoisomerase 1 cuts one strand of DNA to relieve its tension. Topoisomerase 2, known as a DNA gyrase in bacteria, cuts both strands to manage supercoils. Fun fact, DNA gyrase is the reason bacteria can twist and compact their circular DNA efficiently. Without it, bacterial DNA would be too bulky to fit. Now, who remembers the name of the enzyme that helps the bacterial DNA? Oh, it is DNA gyrase. Let's move on from the twisting of DNA to wrapping, specifically how DNA is packaged using proteins. The smallest unit of packed DNA is the nucleosome. It is like a bead made of DNA wrapped around 8 histone proteins. About 147 base pairs make one wrap. These beads on a string are the first level of DNA packaging.
Crazy.
What do you think? Can a single strand of DNA float freely in the nucleus? The answer is no. It would get tangled and be too long to manage. Speaking of nucleosomes, DNA folds into thicker structures. The 30 nanometer fiber is a coil of nucleosomes. This fiber loops into domains attached to a protein scaffold. Finally, during cell division, it condenses further into chromosomes. This step-by-step folding lets the cell keep DNA compact yet still accessible. Chromatin can be either 𝐖𝐞𝐥𝐜𝐸chromatin or heterochromatin. 𝐖𝐞𝐥𝐸chromatin, it is loosely packed and it is active in making proteins, while heterochromatin, it is tightly packed and is usually silent. Here's a cool detail. Think about this. If a gene needs to be used often, would the DNA around it be packed tightly or loosely? The answer is loosely, so it would be 𝐖𝐞𝐥𝐸chromatin. So why does this matter in the cell? Well, DNA supercoiling and packaging aren't just for one space saving. They are essential for DNA compaction and storage, gene regulation, replication, and transcription. Now, DNA compaction and storage is getting that 2 meters of DNA to fit. In gene regulation, it is controlling what genes are on or off. and DNA replication and transcription, making sure enzymes can read and copy the DNA when needed. Without these systems, cells wouldn't function properly. In summary, DNA supercoiling twists the DNA to reduce space, and chromatin packaging organizes it using nucleosomes and higher order structures. These processes are essential for storing, protecting, and accessing genetic information. And that wraps up our presentation on DNA supercoiling and chromatin packaging. Thanks for listening, and we hope that the next time you think of DNA, you imagine it not just a double helix, but a masterfully organized, coiled, and compacted system. This is Faye from the G-nius Review Center, and I hope to see you next time.